All right, so the first half of this course, right, we concentrated on the structure of the microbes, right? We did all the staining, we looked at them on the microscope, all that good stuff. The second half, you may have noticed that we're more talking about how they function, right? What can they do? What do they do, right? So it's almost like anatomy and physiology, right? But for microbes, right? And we're on the physiology part, right? So structure, things like, does it have a capsule, right? Which we've repeated a little bit with our unknowns, right? We've looked at structures and stains and stuff again. And then now we're really asking, what can they do? So we're asking questions like, can they ferment glucose, right? And you asked that question actually a couple of times today about your organisms. So today we already performed the methyl red and Vogue's Proskauer tests. We've recorded our results for phenol red broths, and we're going to talk about those in this lecture. You recorded your results for citrate. We've done the catalase test and recorded our results. And at the very end, we're going to talk about NRO-tube, a system that I bet you wish we had, right? Because we could have done all of this in one fell swoop for some of the organisms that you guys have. Not all of them. So these are differential tests, right? Based on their differential biochemical abilities, their functions, right? Their physiology for your organisms. So for next week, you'll notice and everyone picked up your homework for next week, right? Next week, we're going to look at your results from the plates that you inoculated today, which are the mannitol salt agar plates, the pink divided plates, the EMB plates, and then I had a few of you guys do the blood agar because it'll help confirm your organism. So we already did that. And then we just did as a backup a new NA slant or TSA. So this is the media that we'll look about next time. And this is the media that your homework is about, right? Reading about these 3 different types of media. And this is the last of the data that you're going to collect on your unknowns, although you have enough data today to actually figure out what you've got. So we've already done this, right? And so for each of these tests, the instructor is going to run negative and positive controls. Why do we do this? Yeah, to make sure that things are working properly, and also, especially for you guys, if this is the first time you're doing it, if you didn't have your book to see the pictures and stuff, to know what positive and negative controls look like, to have a comparison, right? What does a negative result look like? What does a positive result look like? So, we followed these steps already, right? This was my pre-lab, actually. So there's our bubbles, right, of O2 gas from the catalase test. So, a little review. Redox reactions, right? Oxidation, reduction. What do we remember to remember what is what? What's saying? Did I tell you guys last time? Oil rig, right? Oxidation is the loss of electrons. Reduction is the gain of electrons. So, electrons don't disappear, right? They just travel, right? So if one molecule loses it, right, is oxidized, somebody else gains it. So oxidation goes with reduction. So you always have what we call redox, right? Reduction and oxidation reactions. So all of these reactions and stuff that we talk about involve the movement of electrons. Usually they're also accompanied by protons. Basically, a proton and electron is what atom? It's the simplest atom. It's number 1 on the periodic table. Hydrogen. Hydrogen. So a lot of times you're going to see hydrogens moving. So in the case of NAD, which is a modified nucleotide, it has nicotinamide here. That's the molecular component here. And then it's basically 2 nucleotides, right? Phosphate group, the sugar, and here the adenine, where instead of adenine here, it's replaced with the nicotinamide. So that's what NAD stands for, right? So this nicotinamide adenine dinucleotide. That's a mouthful for you, right? Hence why we abbreviated NAD, right? But this is what this molecule is. And what this molecule helps us do is carry electrons. We commonly refer to this molecule as an electron carrier, right? Because it will pick up electrons from molecules, right? And so a lot of times when it picks up the electrons, it also will pick up a hydrogen atom. So it goes from NAD+, right, which is positively charged, to NADH, which does not have a charge. It picked up hydrogen atom, right, and protons, and that's why it's no longer charged. And that's also why there's an H added to the abbreviation. It actually picked up a hydrogen atom, along with two sets of electrons, right? So again, hence why it's called an electron carrier. So what are the three main pathways in respiration? What happens first? We got to break down a glucose molecule. What do we call that process? Glycolysis. And then it's going to be converted into another molecule that goes into what cycle? The Krebs cycle, or citric acid, or TCA cycle. And we're going to pull a whole bunch of electrons from glucose, right, in the Krebs cycle, and some from glycolysis. And then where do those electrons travel to in order for us to generate another carrier molecule that basically just carries energy for us? So where do the electrons go? The electron transport chain, right? Very important. So glycolysis, to break down to 2-pyruvate, right? Or pyruvic acid, right? These are similar molecules. Most of us use the names interchangeably. We're not going to get into the big-time chemical differences, right? Keep it simplistic. Then there's a conversion step, or a gateway step, where the pyruvate combined with acetyl-CoA, right? The coenzyme A to form two acetyl-CoAs, which actually enter into the Krebs cycle. In that process, notice carbon dioxide is given off as a byproduct. So then the acetyl-CoAs enter into the Krebs cycle. In the Krebs cycle, we're going to, again, get rid of the carbon in the form of carbon dioxide. And then the electron carriers are going to carry the electrons to the electron transport chain, which is embedded in the membrane. It reduces the coenzymes, right, from steps 1 and the gateway step and the Krebs cycle, right? All this is going to the electron transport chain. So those electrons are eventually picked up by what molecule? If it's aerobic respiration, oxygen, and we form water. And again, notice hydrogen ions are involved, right? Protons. So they usually follow their electron buddies. Ultimately, carbohydrates like glucose, which we're going to look at a couple of times today, are oxidized to carbon dioxide, right? And we store that energy of breaking those bonds in ATP. So let's look at this a little bit more detailed. So here's good old glucose, which is a carbohydrate. Carbohydrate means for each carbon molecule, you have a water molecule. That's where they get their name, right? They're carbon and water, basically, is what sugars are. So at the substrate level, so at the enzymatic level, we can actually generate some ATP in glycolysis. Right, this is the Ebden-Meyerhof Preris, which usually gets left off. Ebden-Meyerhof. There's another type of glycolysis, but this is the most common. Here's that electron carrier, right? NAD becomes NADH. It's picking up electrons, right, from the glucose molecule. And then we're splitting, right? Glycolysis, lysing, we're splitting this carbon molecule, right? This is just a simplistic view of the overall of this process to form 2 pyruvic acids, right? So a 6-carbon molecule gets reduced to a 3-carbon molecule, 2 3-carbon molecules. So no loss of carbon as of yet. We've maintained carbon. But we do generate 1 ATP and an electron carrier. Sometimes 2 electron carriers. In the Ebden-Meyerhof, it's a different electron carrier. This one, notice it's NADPH. It has an additional phosphate. So some organisms use that type of glycolysis, of breaking down glucose. So then we have our gateway step. We're going to take a pyruvate, and we're going to actually, in this case, as we add the coenzyme, we're going to lose a carbon dioxide. And we're going to also pick up more electrons by the electron carrier NAD to form NADH. Right? So we switch out the carbon, right? So now it's carbon dioxide. And then the movement of the hydrogen ions and the electrons. So acetyl group, right, is why it's called that. And so then we're going to enter into now the Krebs cycle. So here with the Krebs cycle, acetyl-CoA combines with oxalisate, which is a 4-carbon sugar, which forms back a 6-carbon sugar, known as citric acid, right? That's why sometimes the Krebs cycle is referred to as a citric acid cycle, and also referred to as citrate, which we're going to talk about again today, right? And so also citrate has 3 carboxyl groups, so that's another reason why it's referred to as the tricarboxylic acid pathway. Again, it's referring to what citrate is. And so unfortunately lots of names, right? But we can break down this molecule, and as we do that, of course, we're going to release the carbons in the form of carbon dioxide, right? So we're going to remove these carbons that we added, and we're going to release them as carbon dioxide. We're going to take away a whole bunch of electrons, and we're going to use electron carriers to do that. So you'll see we'll generate 3 NADs to NADH, and a new electron carrier we haven't talked about yet, FAD, is also used in this process. Notice the oxalisate is regenerated, right? So this is a cycle. It just keeps going around and around each time an acetyl-CoA is added in, right? We break down the acetyl into carbon dioxide molecules, and we take electrons. And we also generate some GTP, which is very similar to ATP, it just has a different nitrogenous base, but storing that energy in that phosphate bond, that 3rd phosphate bond that they make. And this is right at the substrate level, right? So right at the enzymatic level. We're generating a little bit of energy, right? And of course, we're reducing, they're gaining electrons, right? These electron carriers are picking up electrons. And the carbon dioxide is actually oxidation. Oops, there's our important note. So the citrate test detects organisms that can take up citrate from the media rather than synthesize it. So we're not saying, does the organism have the citric acid cycle, right? Can it make citrate? We're saying, can it take citrate from the media into its cell? Okay. When it does that, it's also going to metabolize and form ammonia. And the ammonia is actually what we use as an indicator for that test and causes the pH change that you see in that tube. And basically they're taking in citrate, right, and basically doing the reverse. They're breaking it down into oxalate and citrate for use to build other molecules. So electron transport chain. We have 2 electron carriers, NADH and FADH2. When they donate their electrons, which are embedded in a membrane, for eukaryotic cells, where is this happening? What membrane where? In the mitochondrial membranes, right? For prokaryotes like our bacteria, it's happening in the cellular membrane, right? The cell membrane. So these proteins are embedded, right? And they pass electrons down to the final electron acceptor, which is, if it's aerobic respiration, oxygen to form metabolic water. In this process, right, of moving these electrons down this membrane, oops, I went the wrong way. Protons, right, hydrogen ions, just the proton, are pumped across at 3 spots for NAD and only at 2 spots for FADH. So we're moving protons across the membrane. So as we said, it happens in the mitochondria for eukaryotes, right? It happens in the membrane, the cellular membrane for prokaryotes. So once we pump those hydrogen ions either outside the cell or into the inner membrane space in the mitochondria, those protons actually travel back through a special protein port in the membrane. This generates the energy necessary to add the phosphate to NAD to form, not NAD, ADP to form ATP, right, inside the matrix of the mitochondria. This is referred to as oxidative phosphorylation because we're using electron transport train, right, the movement of the electrons to generate the proton pump that drives the ATP synthase. So oxidative, and again, refers to the loss of electrons in this case. So the problem for aerobic organisms, or organisms that can respire aerobically, is this electron transport chain. Sometimes electrons jump ship where they're not supposed to, right? And so here, if two electrons leave the electron transport chain here and add to oxygen, it forms a superoxide radical. And this is a highly reactive form of oxygen. It can be very damaging. Okay, so we don't want this to happen, but we can't stop it from happening. But we protect ourselves instead from these toxic forms of oxygen with enzymes that break down these toxic forms. So the other toxic form, anyone know that can form? Other than superoxide radical, we actually used it today as a reagent. Hydrogen peroxide, right? So with the addition of some protons, we can form hydrogen peroxide, which is also very destructive. Not as powerful as superoxide, right? But pretty mean. So our catalase test tests for one of these enzymes, the one that breaks down, of course, hydrogen peroxide, which is why we use hydrogen peroxide as a reagent today. It's a redox reaction, right? Loss and gain of electrons. So hydrogen peroxide, and this is the important formula you want to write down for your unknown report, right? Breaks down into water and gaseous oxygen. So remember, the bubbles that we see is the oxygen gas being released as hydrogen peroxide is broken down into water and gaseous oxygen, right? So if you're using a electron transport chain, right? You're doing aerobic respiration, so you definitely need oxygen. You're living in the presence of oxygen. You're using oxygen. So guess what you're doing? You're making superoxide radicals. You're making hydrogen peroxide. So do you need catalase if you're using aerobic respiration? Yeah, because you're making hydrogen peroxide, are you not? You are. So if you don't break that down back into safe oxygen and water, you will die. So all of our organisms bubbled except Grace's, right? Because is Grace's organism using aerobic respiration? No. So guess what? She doesn't need catalase. She's not making hydrogen peroxide. Make sense? Okay. So, summary of catabolism, the breakdown of glucose, right? We're going to generate carbon dioxide, right? Couple of electron carriers, and even some GTP and ATP at the substrate level. But all these electron carriers, remember, got to go to the electron transport chain. So how much do we get for our glucose? 3 ATPs are produced for each NADH, because remember, 3 sets of protons went across the membrane. For FAD, we're only going to get 2 ATPs, because only 2 sets of protons got pumped across the membrane. So therefore, for aerobic respiration, which is glycolysis, the gateway step, and the Krebs cycle, and electron transport chain combined, theoretically, you could yield in bacteria 38 ATP per glucose molecule. Eukaryotes, however, only produce about 36 theoretically, because they have to expend some to move across the membranes of the mitochondria. Glycolysis happens outside the mitochondria, but the Krebs cycle happens inside. So you lose energy as you transport the pyruvates into the inside of the mitochondria. So 2 ATPs and 2 GTPs, essentially 4 ATPs, are produced at the substrate level. All the rest, right, 34 of that total are produced by the electron transport chain. So if you can't utilize the electron transport chain, say you don't have any oxygen available, right, you can only generate 4 ATPs from just being able to do glycolysis. That's it. Still with me? All right, so what do you want? 30s? Let's say you're a bacteria. Do you want $38 an hour or 4? Yeah, I'm going to take the 38 over $4 an hour, right? But again, you can only do the 38 if you have what? Oxygen, so that you can use the electron transport chain. If you can't use the electron transport chain, you're stuck with just glycolysis, just breaking down glucose. Y'all still with me? Right? So that kind of sucks energy-wise, right? We've looked at the math. Now, of course, these are theoretical yields. Actual yields are a little bit lower. And energy, therefore, not as awesome as you think it would be. These are some cool videos that go over, right, electron carriers and electron transport chain. So, oops, this is a very important question. If an organism lacks the electron transport chain, how does it recycle the reduced coenzymes? Because remember, in glycolysis, we do generate 2 NADs, right? If we have no place for those electrons to go, how do we get rid of them so that we can pick up electrons from the next glucose that's broken down? Those electrons have got to go somewhere, right? That carrier does just that. It just carries them. It picks them up and is supposed to bring them to electron transport chain. No oxygen. Can't go. So you got to dump the electrons off somewhere, right? So you dump them back to what you broke down, right? So you're going to give them back to pyruvate. In that, you're going to form organic molecules, acids, alcohols, and gases. This process we collectively refer to as fermentation. When you break down glucose without oxygen, without being able to utilize or without having an electron transport chain, you got to add those electrons back to your breakdown products. Now, of course, we don't just add it to pyruvate and that's the end of the story. They do additional processing sometimes to these molecules, and that's where we get these acids, these gases, and these alcohols, which we love so much, right? We love fermentation. So good old pyruvate, right? Breakdown product from glycolysis. Problem is we've got some electron carriers. So one of the things we can do is remove more carbon in the form of carbon dioxide to form formaldehyde, which then we can add the electrons back to this, and it forms ethanol, like I said, one of our favorite alcohols, right? Produced by brewers and bakers' yeast, good old Saccharomyces cerevisiae. Or we can remove carbon dioxide and form formaldehyde and instead add a pyruvate molecule to the formaldehyde and form alpha-acetyl lactate, an acid. We can remove carbon dioxide from it and add protons and form acetonin. Acetonin you should have wrote down on your homework, because if you are positive for which test, your organism produces acetonin. Acetonin is then converted by adding the electron carriers to 2,3-butanol, which is an alcohol. And we test for the ability to make 2,3-butanol by testing for the presence of acetonin and the Voges-Proskauer test today, our VP test. Right? So if yours turned red, which means that you had acetonin, which means your organism can do this specific type of fermentation. It can do 2,3-butanol fermentation when given glucose. And glucose was in the MRVP media we used. So other alternatives, right? We can add the electrons right back to provate, like I said, and in that case we form lactic acid. And this happens in our own bodies, right? Sometimes when we don't have enough oxygen during exercise. But this is bad, right? Acids can be bad, and so you don't want this to happen in our body for too long. So lactose, lactis. This bacteria actually is what converts, milk products by this type of fermentation to form buttermilk and cheeses and yogurts. Lactobacillus species, these are essential for making and fermenting the sugars found in different food products to form different fermented foods like kimchi and sauerkraut and sourdough bread. And again, it's that acidic content that makes these things sour. Acids are sour to us. Other lactobacilli are crucial in the female vagina for maintaining, again, that acidic low pH, which will help keep yeast under control. Our red blood cells actually lack mitochondria, and they actually produce high levels of lactic acid. Lactase dehydrogenase, right, is an enzyme that actually breaks down lactate. And so when our cells are damaged, this is released, and this is actually one of the tests they can do to determine destruction to muscles, lungs, heart, right, different things. If you have high levels of this enzyme being released, it means you have a lot of cell death, and that's not good news. Right? But this is one of the ways that we protect ourselves. So another thing that can happen is more NADH can be added to the lactic acid to form propionic acid. Most organisms don't do it this way, though. They instead add carbon dioxide to the pyruvic acid to form oxalate, then convert that into succinate, right? And this is, again, we're reusing parts of the Krebs cycle. And then that is converted into propionate. So it's all about, right, getting rid of those electrons. And we can do it a whole bunch of different ways, right? So if you guys look in your book, right, in the appendix 5, it lists all the different pathways, right? We're not memorizing pathways. The important take-home message here is that we're going to be giving back those electrons to pyruvate from breaking down of glucose and glycolysis. And we're going to form alcohols, acids, and gases, and ketones. Right? So, lots of gas, right? Acids. And here's the other gas, hydrogen gas. Hydrogen gas is unique to fermentation. It does not happen, but remember, we give off carbon dioxide in aerobic respiration, right? That's how we're getting rid of the carbon from glucose. In our phenol red broths, you don't know which gas it is. It's either hydrogen gas or carbon dioxide gas or both. But you know it's gas being produced. Our methyl red test is specifically for acids, right? It's a pH test that we do, right, for lots of different acids. So anaerobic bacteria, as I said, produce hydrogen gas. That is unique. And so when animals die and they bloat, one of the common bacteria that are spore formers found in the environment is Clostridium perfringens. This is also causes a condition if you have serious wounds called gas gangrene, right? Because it will literally eat away at your tissue. But this one also, when it ferments and eats away at your dead tissue in a wound, it produces a lot of gas. So you'll actually get really bubbly, frothy discharge from it, and that's why it's called gas gangrene. And again, why they will bloat because of the gas produced. Hydrogen gas is lighter than air, but unfortunately very flammable as well. So the Hindenburg is probably the most famous of the airships that blew up because they were using hydrogen gas. And then, of course, they switched to using helium, which is not flammable. Most people know about the exploding Hindenburg, huh? Even though that was in the 1930s. Can you imagine that? That's so long ago now. So, possible products, right? What are the general products? What do we say? 2 A's, acid, our favorite, alcohols, and gas, right? So, a bunch of different acids can be produced, a bunch of different alcohols can be produced, and ketones, and gases. Methanol, right, is not in the figure in your book, and it can be toxic to humans. It interferes with our electron transport chain, and if you drink it, it could make you go blind because your optic nerve does a lot of aerobic respiration, and if you shut that down, then you're not going to be able to see. So the methyl red test and the Voges-Proskauer test. So which one tests for mixed acids? Which one was an acid test? Which one? The methyl red, because remember it's a pH test. Remember methyl red is a pH indicator. So that was the reagent we used, right? We used methyl red. It's a pH indicator. It's important that you know that. What do the products do to the pH of the media? So they're acids. What happens to the pH? It goes, if you have acids, pH goes down, down. pH goes down, lowers the pH. Right, so our good old hydrogen ions, protons. And as I said, they're very significant. They tend to follow around their electron buddies. But if they lose their electron, which they aren't all that attached to, right, they'll readily give up their electrons to other molecules, then they form the proton, right, the hydrogen ion. And that's what the measure of pH is measuring, right? It's the partial pressure of free hydrogen ions, protons. The problem is that protons can also bind to other things, right, and create problems. So if you have too much of them, it can be a problem. If you don't have enough of them, it can be a problem, right? You need to maintain some level of balance. So remember, what's balanced pH? What's the balance of hydrogen ions? What's that number? For neutral pH, where we have that balance in a solution. What's that magic number for pH? It's neutrality. 7, 7, 7. So 7 is our midpoint, right? And it's the negative log of the concentration of hydrogen ions, right? So what we find out is that when you have a low pH, because it's the negative log, right? When you have a low pH, that actually equals high hydrogen ion concentration. When you have a high pH, or a basic or alkaline condition, then you have a low number of free hydrogen ions, right? You don't potentially maybe have enough. Neutrality is very important. For us in our bodies, we in our blood must maintain a range of 7.35 to 7.45. You go acidic, right? Is that when you convulse or you slip into a coma? I always forget which way it goes. I don't know my A&P2 all that well. But basically, if you go too low or too high, right, these are the two options. You either convulse and can't breathe and die, or you slip into a coma and die, right? Acid-base balance is very, very important, right? So we use lots of different pH indicators in this class, right? We've used crystal violet as a dye, but crystal violet is also a pH indicator. Phenol red is what's in our phenol red tubes, right? It's a pH indicator. We could tell the pH change, right, whether it was low or high based on the two colors, whether it was yellow or pink. Methyl red, right? Again, it was a pH indicator. Again, it told us if it dropped to a particular low pH, right, a 4.4 or lower. Notice the different range for phenol red. It's not quite as acidic, right? Just has to go a little bit acidic, 6.8, and it'll turn yellow. Where methyl red, you really had to have a lot of acid. This is very acidic, right? 4.4 for it to stay red. Citrate test. This test helps us detect an alkaline environment. So the pH, when it gets above neutral of 7.6 or higher, it turns blue. Hence the name bromelain blue. So fermentation that produces 2,3-butanol. Which test? The VP, the Vogt-Proskauer test. What reagents did we use? We used two, didn't we? We used Barrett's reagent A and B, alpha-nethaphenol, and potassium hydroxide, which remember is a very strong base, which is why we definitely wanted to wear gloves. I will make so you guys spell these guys on the test, but you do need to be able to recognize the reagents that we use. What intermediate product is being detected in this test? We don't detect for the presence of 2,3-butanol, do we? We detect for its intermediate, with this what? Acetonin. Not to be confused with acetone. Different molecules. Acetonin. So this is what happened, right? We gave your cells glucose, they broke it down by glycolysis, but they fermented it. They didn't go into the Krebs cycle or use electron transport chain. They converted it into acetonin. Then we took a sample, right? And if it contained acetonin, it binded with the alpha-nethaphenol. Then we added oxygen, remember, with our second reagent, the potassium hydroxide. This will form an diacetyl that will bind with the guanine in the media, and it will produce a red color if the acetonin is present. Right, so we're detecting acetonin, right? Remember, we needed to shake the tubes really good at that second step to add the oxygen. This is a chemical reaction that has to take place, right? So that's why we checked it every 10 minutes. We didn't even have to go a whole hour though, right? All the positives had turns positive in by the first 20 minutes. This copper color that you'll sometimes get is not positive, right? You want a red color for positive. So for methyl red test and the VP test, what color indicates positive? Red for both of these guys. Just worked out that way. So notice this one's kind of a weak positive, but it's positive. There's red there. So methyl red test, again, just pH test, where VP test, a chemical reaction has to take place in order to form this molecule that produces the red color. So there's a better picture of the tubes. So these are definitely positive, right? This one's just weakly positive. That one, no reaction, negative. So would a false negative result for the VP test more likely be contributed to poor sensitivity or poor specificity of the test system? Sensitivity. Why sensitivity? What has to happen? A reaction to produce what? A product that we can see, right? That we can see. And again, you've got to produce enough red that you see it, right? You have to have enough there. So that's sensitivity. Specificity would be like if the reagents bind with something other than acetonin, right? Then we wouldn't have a very specific test. And of course, then we wouldn't really want to use that test if it wasn't specific, right? But there is a problem with sensitivity here, right? In that we have to produce enough color that we can actually see it. So, when acetonin is quickly converted to the end products, there'll be very little in the intermediate form for us to detect. There are other circumstances where the concentration of acetosine might be low. For example, perhaps acetonin lingers around for a long time in a particular species of bacteria, but not much acetonin is generated in the first place, right? Maybe because you didn't put enough bacteria in the tube, or the growth temperatures weren't optimum. That's not an issue for us, too, because typically you would read these or run these tests 2 days later. Ours have been sitting around for quite some time, right? We did these weeks ago. So we shouldn't have had any false negative results, and we didn't. Everyone who came out who was supposed to be positive was positive. So why were you told to shake the VB tubes after the reagents were added? What else did we need to add? Right? We needed to add oxygen for this reaction to proceed. So the methyl red test, remember, is just a pH test. So we just added our pH indicator, a couple of drops of phenol red, and we read it immediately. Red color indicates that very low, 4.4 or lower pH. Yellow or orange is a higher pH than 4.4 and therefore would be negative for this test. So methyl red, we're looking for organisms that do what we call mixed acid fermentation, where they produce a lot of acids, right? So you can see here, right, for instance, this example of E. coli, it can ferment glucose and produce lactic acid, carbon dioxide, hydrogen gas, ethanol, acetate, formate, and succinate. And that was the positive control I used for this test. Gave us a nice positive result. Why is the methyl red test read immediately, but we had to wait for the Vox-Proskauer for about 60 minutes potentially? What's the methyl red test? What is methyl red? It's just a pH test, right? This happens immediately, right? Within seconds. The VP test, we're waiting for what to happen? A chemical reaction, and these can take time, right? So we have to allow the time for that to occur. We can't just read the results right away. So some microbiologists recommend re-incubating organisms producing methyl red negative results for an additional 2 to 3 days. Why do you think this was done? More time, you're going to produce more what? Acid, right? So more likely to get a positive result instead of a false negative. Again, ours were inoculated a long time ago, right? The only problem is we still had some false negatives, right? So I don't know what was going on there. So we'll investigate that. All right, so that's our VP test. Now we move on to phenol red. So phenol red were the tubes with the little tubes inside called the Durham tubes, and they were there, right? And we use a code system to code these tubes based on whether acid is produced or alkaline products and whether gas is present or not. There are lots of different sugars that you could test for, right? So notice this huge list of sugar and sugar derivatives. Anyone know which sugar we're testing for the ability to break down?
Lactose?
Nope. Glucose. Glucose. Lactose, sometimes, some labs do that. But we do glucose. So you'll notice the formula in your book, right, as I said, right, does contain glucose. Phenol red is the pH indicator in these tubes. It allows us to determine the pH change. The protein source in the tube is a pancreatic digest of casein. Casein is the most prominent protein found in milk, and actually what gives milk its white color. And so it's partially digested. Some other media, like the TSA that you guys use, contains partially digested casein. And it's digested using pancreatic enzymes, so a lot of times it's written tryptone or tryptase. So amino acid source, right, and carbon source, right? You always need a nitrogen, an amino source, and a carbon source for your organism. So they have both in this media. And again, depending on which sugar you want to ask the question for would determine which sugar you would put in that media. and we use the glucose. So what's the common byproducts of fermentation? What's the A's? Alcohols and acids and gas. And gases are just hydrogen and carbon dioxide. Which one of the three can we not detect with the phenol red broth test? Can we detect gases? Yeah, that's why we have the Durham tube, right? Can we detect alcohols? No. No. Can we detect acids? Yes, because what's phenol red? pH indicator, right? So we can tell a change in pH. We can tell if acids are produced. We can't tell alcohols, right? The only test that we do that tells us anything about an alcohol being produced is the VP test. It's the only test that deals with alcohol from the production from fermentation. So what's the pH indicator in this media? It's the name of this test. Phenol red, right? Phenol red is going to turn yellow at acidic pH and not very acidic, right? Just 6.8. It'll turn red at higher pHs at basic and magenta at 8.2 or higher. So what color does the media turn if acid products are generated? Yellow, right? So here's a tube, right? You can clearly see the yellow color, right? So you know that's acid production, but you don't see any bubble in the Durham tube, so it's minus for gas, right? Negative. What color does the media turn if alkaline products are generated? Right, that magenta pink color, right? So you can see this nice pink tube. And we code that K. What does a Durham tube help detect? Gas, right? So you see in this one, you see that bubble, right? So that gas bubble, right, that's trapped gas in that inverted tube that we call a Durham tube. So we would code that A because it's acid, right, and G for gas. And again, you're going to get acid, right, because fermentation is happening. And sometimes you'll get gas. Not all of them produce gas when they ferment. So only some of you guys had some gas bubbles. Actually, I think only one person that's here today. So if glucose is fermented, what happens to the pH of the media? So if it's acid, the pH went down. Still with me? Okay. So peptone, right, a mixture of proteins and polypeptides, is a media component that some organisms can use as a carbon and energy source, right? So if they don't utilize the glucose, they're going to use the protein instead. When they do that, they're going to produce ammonia. Ammonia does what to the pH? It raises it. And so again, it's going to create a color change. In this case, it's going to be that pink magenta color. And we code that K for alkaline. Because of course, alkaline and acid both start with A, right? So we got to go with the next letter. We got to go with K for alkaline. Right. So, and of course, remember, if pH increases, hydrogen ion concentration actually decreases. Right, so alkaline ammonia. It actually absorbs the hydrogen ion. See how it becomes positively charged, that ammonia? And that's because they're breaking down the amino acids, right? So an amino acid has a carboxyl group and an amino group. If you remove the carboxyl group in the form of carbon dioxide, you're decarboxylating it. If you remove the amino group, right, and form ammonia in solution, then you're deaminating, right? And that's what these organisms that are turning pink are doing. And again, they're going to add back the electron carriers to it, and they'll form other types of molecules. In that case, when they're adding electrons, remember, actually they're removing electrons, so they're oxidizing. But this becomes reduced. It gains the electrons. The electron carrier gains the electrons. And this can go to the electron transport chain to generate energy, right? So they're breaking down, instead of breaking down sugar, right, to generate electron carriers, they're breaking down the amino acids. They're deaminating them, right? Here's decarboxylation, right? Here's another deamination. Oh, and the reason why these two are mentioned is the last test system that we don't do anymore that I'll show you guys about. You can test if they can actually break down this particular amino acid or this one, orthonine or phenylalanine, if they can break it down. They can deaminate them. So when microbiologists do these tests, sometimes they'll use base broth, phenol red base broth, which just has the protein in it and not a sugar added as a control set. We did not do that. So we used media that contained both glucose, right, a sugar source, and the natural protein source in the base broth. The base broth would contain protein only. So what happens if the bacteria eats the glucose? How do we know it ate the glucose? What's going to happen? It becomes acidic, right? So they produce the acid byproducts and it becomes acidic. How do we know that tube is acidic? It's yellow, right? The pH gets lowered and it gets turned yellow. What happens if the bacteria eats the protein? The pH is going to go up, right? And it's going to turn that pink or magenta color. What happens if the bacteria eat all the sugar and then begin to eat the protein? So this is why these were in the refrigerator, right? And why I gave Jessica a special tube. Because hers ate up all the sugar in 2 days, right? In 48 hours, in 2 days, it ate up all the sugar and then it started to eat the protein. So what did you see in her tube? You saw yellow, right? But then we also saw orange at the top because the pH went down, which was yellow, right? But now it's coming, the pH is coming back up, right? Orange is neutral. If you let it keep going, eventually it'll turn pink, right? So it really depends on where you catch it, right? What would the final pH be? It's kind of hard to determine. Depends on when you look at it. Now for protein-only tubes, what's the only possibility that can happen? Pink, right? That's the only thing that can happen, right? If you have just protein, if you don't have a sugar, you can't ferment. You can't get acid. You can't get gas. Is everyone clear on that? It's an important point. Okay. So there is some of the things that happen for two of the organisms we have, right? Where you get that yellow, they ate the sugar, right? Because you see the yellow, but that now they've moved on to protein, so that's why you're seeing this orange layer. What's different between these two tubes, though? Right, one of them has gas. And so the good news about that one, which Jessica has, and which we would code this AG with weak K, right, where it's starting to eat the protein, is that if this tube went too long, and say it was orange, you would know something happened, right? Because that gas bubble will indicate, well, fermentation must have happened, right? At one point, this tube was yellow. But this guy really loves his sugar and ate it really fast. This one, on the other hand, notice no bubble. So if this tube goes, right, longer, you might not notice that acid happened first, that fermentation happened first, and then it started to eat the protein. So it's one of the main reasons why I took these out of the incubator, why they weren't near racks, and I refrigerated them, was to slow the growth so that you can see these transitions that happen for these two organisms. So the bubble one is a good indicator that it ate the sugar first, right? Because you only get gas with fermentation of sugar, not with protein digestion. So the good thing with that one is if you ended up with, say, an orange or a pink tube, and you saw that bubble, you would know, oh, fermentation happened first, and then now it's eating protein. Where with this tube where you don't have any gas, right, if that tube goes too long and it turns orange or pink, you might code it wrong. You will notice that it actually ate the sugar first. Yeah, it just ate the sugar and it didn't produce any gas in the fermentation of that sugar. If it's just a yellow tube, right, no gas. Which most of your organisms, that's what happened, right? And then some of you guys had ones that just ate the protein. So in this interpretation chart, again, we use our coded system. And remember, for these tubes right here, this is the base broth with a carbohydrate. So remember, you're going to see all the possibilities in these tubes, right? All the possibilities I have in the racks in front of you. If you were, which we did not do, going to do a set of controls of just the base broth, this only contains protein, no sugar. So what's the only possible reliable outcome that could happen? Or there's actually two outcomes that could happen. If you give the organism protein, it could eat it, right? And turn pink. Or it could do nothing at all. That's it. Can you get acid or gas production from protein? No. Right? Only from the fermentation of a sugar like glucose. So when they ask you about the reliability of the tests that they run in the chart, you're really looking at that second tube, right? Is that possible for the second tube, for it to have the result there? So all of these are possible. We're just basically learning our code, right? So with sugar, can you get acid and gas production? Absolutely, right? It could ferment the sugar, produce acid and gas. You've got a yellow tube with a bubble. If you have a protein-only tube, could it be yellow with a gas bubble? No! Right? This would put your whole experiment in question, right? This would not be reliable at all. How the heck did that happen? Right? Somebody screwed up is what happened, right? Can you have nothing happen, no fermentation, if you have only protein? Of course, right? So what would a negative tube look like if the bacteria didn't do anything with the protein? What color would that tube be? Yeah, the original color, right? Which is around an orange to reddish color. So this is possible, right? They could choose not to do anything. Could you have an organism eat the sugar, right, ferment it, produce acid and gas, and then when given the protein-only source, for it to digest the protein and produce a pink tube? Yes, whose organism would probably do that? Jessica's, right? So this would be a reliable result, right? If we gave it sugar, it's going to eat it, and it's going to produce acid and gas. If we give it just protein, it's like, okay, all right, I can deal with that, right? I'll eat that. I'm good. Right? This next one, could we have this result in a protein-only tube? Could we have acid and no gas? No. That would not be a reliable result. Okay, so K, remember, is the breakdown of the protein, and it produces alkaline products, which turns the media pink. So if we then, of course, give them just protein, will they do that same thing again? Sure. Here they had a choice, right? Sugar or protein. They said, I'll take the protein bar. Right? Smart bacteria. Right? I'm more like Jessica's bacteria, right? When given the choice between protein and sugar, I go right for the sugar. I'm eating the Snickers bar while the hamburger's grilling. I can't wait. Right? What happens if they do alkaline products, right, and the choice between sugar and protein, but then when you give it protein, it doesn't do anything? Is this a reliable result? No, it should eat the protein again, right? Why didn't it? What's going on? Can you get an organism that ferments the sugar when given the choice, but no gas is produced? And then when given protein only, eat the protein? Yeah, that's the other one that I showed you guys, right? If given sugar, it's going to choose sugar first, right? And it's going to ferment it, but in this case, no gas production, but acid. So, and when it runs out of sugar, it'll start eating the protein. So if you just give it protein, it will eat it. Can you get acid and gas production in a protein-only tube? No. So therefore, this would not be a reliable result, right? The very first one? Okay. So here they have a choice, sugar or protein, right? They chose protein. Here, they have no choice. It's just protein. So they should eat it again, and it did. This next one's not reliable because given the protein again, it didn't eat it. Well, why didn't it eat it? It should have ate it.
Some bacteria will prefer to eat protein over carbohydrates.
Yes. And those are the people who had the pink tubes. So suppose you inoculate the broth with a very slow growing fermenter. So he's slow, right? So the tube still looks like your control tube 48 hours out. Would this be a false positive or false negative result? False negative, because we said it's a fermenter, right? So it should turn color. But what's the problem? He's slow. He's slow. He's slow. He's slow. Is this a problem with specificity of this test or sensitivity? Again, it's a color change. You got to get enough acid to produce that color change, right? So this would be sensitivity issue. Early formulations of this media contained a small amount of carbohydrate. So that would be a real problem for Jessica's organism, right? Because after 48 hours, it definitely had moved on to eating the protein, right? So we wouldn't know that except for maybe the gas bubble may clue us in that, oh, sugar fermentation probably happened, but why is it not yellow? Well, because he's moved on to the protein. So how could we fix this problem? So what's happened is they ate up all the sugar, right? And now they're starting to eat the protein. So how could we fix this problem? We have a small amount of carbohydrate. Add more sugar. Or look at the tubes earlier, right? Only incubate, say, for a day instead of 2 days. But we actually, of course, put more sugar. Why? I don't know. Why do this for 2 days instead of just 1? I don't know. Okay. So citrate. Remember, citrate test, we're not testing for if they can make citrate, we're testing if they can take in citrate. So basically if they have this permease, which is a protein embedded in their membrane called citrate permease, which of course brings citrate in. Now of course I couldn't find a diagram that showed citrate, so I have a different sugar here. This is sucrose. Same difference, right? We're bringing a sugar in. In order to do that, you need a special protein in the membrane to do that. Not all organisms have citrate permease, right? Those of you guys that were negative for this test, your organism does not have citrate permease. Those of you guys that were positive, it had it, right? So what's the carbon source? Remember, we always have to give them carbon and nitrogen. The carbon source in this case is the citrate. And we always have to give them nitrogen source. The nitrogen source is ammonium dihydrogen phosphate. So again, notice the nitrogen source there. Since this media only has 1 carbon and 1 nitrogen, what happens to the organisms if they cannot take in the citrate to use as a carbon source? They die. They die. This is an evil test. What's the pH indicator in this media? Bromenthal blue. Bromenthal blue. Right, so positive is blue. Negative, it stays green. Right, this stuff starts out green. What color does it turn? If they utilize the only nitrogen source. So they have to utilize the citrate, right? But then they have to use the nitrogen source. I know. I was like, is that right? So it's going to decrease the pH, right? It's going to make it alkaline. We're going to get a blue color. If the media is green and you see growth of the organism in the tube, is this a positive result for the utilization of citrate? Normally, yes, but I caution you guys, special note, right? If you think you're seeing growth in a green tube, you are probably not. You're seeing what's called a heavy inoculum, which means you put too much bacteria in your tube to start with. Growth would be a lot more than what you're seeing. And again, these have been going for so long that only if your tube is blue is it actually positive. Okay? So, yeah, we're out of time. We talked a little bit about catalase in the introduction, so I'm not so worried about that one. So just make sure that you guys read over the rest of the PowerPoint. There's one of them I want to talk about catalase. So give me one more second. I want to find it. Positive. Oh, here we go. What? Would a false positive reaction between the inoculating loop and the hydrogen peroxide be caused by poor specificity or poor sensitivity? So the problem here is if you did this test wrong, you guys, if you put hydrogen peroxide on your slide first and then use the metal loop to add your bacteria to the hydrogen peroxide, you could get bubbling from the metal making contact with the hydrogen peroxide. And this is actually a pretty cool video that Peter found. Metals will react with hydrogen peroxide and cause it to bubble. So if you were to perform this test properly, you would not know if it was the metal or your organism causing the bubbling. So this is poor. This is the one example of poor specificity that we're dealing with in this lecture. It's the one example. The other wise tale that's out there, right? Hydrogen peroxide sometimes people use for wound care. Have I talked about this previously with you guys? No. It's recommended that you don't use it anymore, right? Because it's destructive to your cells as well as the bacterial cells. You can use it to clean your house or your countertops or your jewelry or something like that, but it's not advised on cuts. You are catalase positive or negative? You're positive. We're aerobic, right? Do our cells contain catalase? You betcha they have to. So when you put hydrogen peroxide on a wound, it should always, always bubble, right? Because you are catalase positive. Don't believe the old wise tale that if it doesn't bubble, it's not infected. Instead, know that if it doesn't bubble, your hydrogen peroxide is old and you need to throw it out and get new stuff, right? Because hydrogen peroxide over time, when exposed to light, will turn into water and gaseous oxygen, which is also why it's kept in a dark bottle, to slow that reaction. Okay? All right. So make sure you guys review the rest of this for your quiz next week. Work on answering those questions in your unknown report based on the results you gathered today. And hey, process elimination, see if you can figure out what you've got. We'll talk about that next time. Thank you. It's a lab pencil. Stuff that gets left in lab becomes lab.